Alright, good. So as a general rule, mammalian cells will multiply only if they are stimulated to do so by an extracellular signals that are called mitogens. Alright, so what are mitogens? So they are an extracellular signal to stimulate, right? That will produce the proliferation of cell. Okay, let me just write it down. Maybe over this here. Alright. Oops. Alright, so mammalian cells will only multiply. So mammalian cells will multiply only if they are stimulated by an extracellular. They are stimulated by an extracellular signals, and that are called mitogens. Right? So what will produce, and these mitogens are produced by other cells. So if mitogen is not present, right, the cell cycle will arrest in G1, right? So like the mitogen is responsible to activate the cell proliferation, right? So this phase, entire interface is of first cell proliferation, and if the mitogen is not present, what will happen? The cell cycle will just stay or arrest in the G1 phase, right? And if the cell is deprived of mitogen for long enough, it will withdraw from the cell cycle, right? If like there are certain amount of time, like the cell will wait or it will be arrested in G1, but it will not get mitogen for a long time. What it will do, it will withdraw the cell cycle and enter a non-proliferating state in which the cell can remain for days or weeks, months, or even for a lifetime of the organism. Right? So how it is removed from the cell cycle arrest. So what happens? So to escape from the cell cycle arrest or from other certain non-proliferating states requires accumulation of cyclins, right? As we studied, right? So we need cyclin to activate the proliferation state of this cell cycle. So what the mitogen will do? Mitogen acts by switching on cell signaling pathways that stimulate the synthesis of G1 cyclin or G1S cyclin. So this type of cyclin, the mitogen will act by signaling the pathways on G1 and so like for example, this is mitogen. It will act on G1S cyclin, and this G1S cyclin and other proteins that are involved in DNA synthesis and chromosome activity. So what happened? It started accumulating of these cyclins, right? So the buildup of this cyclins triggers a wave of G1 acidicase, right? So this is G1 acidicase. So this buildup, so eventually when mitogen will keep activating this, giving signal to this cyclin, like it will build up and eventually it will CDK, right? The cyclin dependent protein kinases, and it will act on the cyclin dependent protein kinases, and which ultimately relieves the negative control that otherwise block progression from G1 to S phase. So this is how it is coming out of the cell cycle arrest stage, right? And the crucial negative control of this cell cycle arrest is mediated by retinoblastoma protein. Which protein? So that is retinoblastoma protein, also known as RB protein. So this retinoblastoma was actually initially identified from the studies of rare childhood eye tumor that is called retinoblastoma, that you all know, and in which the RB protein is actually missing or it's defective. But the role of retinoblastoma is what is, so this RB is abundant in the nuclei of all the vertebrate cells. So what it does, this RB protein actually binds to a particular transcription regulators and prevent them from turning on to the genes required for cell proliferation. So leave the cell cycle in the middle. So for example, like if this is RB, so like this shape, and maybe like this I will make it, and it has, I think I will use another one to show you. So for example, this is an RB protein. So what it does, the RB protein, this RB protein binds to a particular transcription regulator. So this is, for example, the brown one that you can see. So that is transcription regulator. So that is useful for the proliferation of cell. So when it binds, it is inactive. Inactive, right? So this is inactive right now. So this is how it controls. So if the RB protein is present in all of these cells, so what it does, it binds to this transcription regulator. And if it's inactive, so the cell cannot proliferate. So what the mitogen do? So mitogen releases these RB breaks. So how it releases? So mitogen, as we see, mitogen acts on G1S cyclin, and then it stimulates the G1S cyclin dependent kinases, and there is a series of complex reaction. And this G1 CDKs, what it do, it phosphorylate this, RB protein. So this, for example, acts on here the activated, right? I will just write it down. This is an activated CDKs. So this activated acts on this protein, and then our RB protein, what will happen? Just, I hope you got this. So this is our RB protein, and it will release, and this is for our transcription regulator. So it releases the transcription regulator, and this become activated, right? So I will just write down here and here, activated. All right, so this will alter its conformation so that it releases bound transcription regulator, which are then free to activate the genes required for cell proliferation. So this transcription regulator will activate the genes that are required for the cell proliferation, and this is how it is done. All right, so this was one mechanism. So there is another mechanism. If the DNA is damaged, how the cell cycle arrests before it goes to the mitosis process, like before replicating the DNA. So I will show you that. What should I remove? All right, I will remove this from here, maybe. Wait. Okay, so the cell cycle control system uses several distinct mechanisms to halt. So there are different mechanisms to halt the progress through the cell cycle if the DNA is damaged. So there are different mechanisms of when the DNA is damaged, it can halt the progression. But one of the mechanism is well understood. So it can do so at variable transition points from one phase to the next one, right? So the mechanism that operates from the G1 to the S phase, which the G1 to the S phase, I will write it down here, G1 to S phase. So this mechanism is well understood. So what happens? So when the DNA damage in G1 causes an increase, what will happen if, for example, here at this point G1 DNA is damaged. Okay, I will write it down here. G1 DNA is damaged at the G1 stage. So what will happen when the DNA is damaged in the G1 phase? So what it will cause an increase in both concentration and activity of a protein that is called P53. So this protein P53, for example, I will just make a square of this protein. Like this is a P53 protein. So this, it will increase the concentration and activity of this P53 protein. So right now, for example, this P53 protein is inactive. Right now, it is inactive. So once the cell cycle see, okay, the DNA is damaged, so we have to stop the cell cycle from moving from G1 to S phase, correct? So what it will do, this DNA, it will start activation of protein kinases that will phosphorylate the G1, the phosphorylate the P53, stabilizing and activating it. So like as this is DNA is damaged, for example. So what will happen? The next phase will be activation. Okay, I think I will use the new one. Right, so what will happen? So activation of protein kinases. So there will be an activation of protein kinases, and what will do that will phosphorylates phosphorylate this P53. Oops, sorry, it will be written like this actually. P53 that will stabilizing phosphorylate this P53, and by phosphorylating it will, so this is inactive right now, it will stabilize and activate this P53. So like right now it is inactive, but after this, so P53, for example, like we'll make this block, and with this, like this is activated, for example, this P53 is activated now. Right, so what will happen? So after activating this P53, so this is a transcription regulator, so that will activates the transcription of gene encoding a CDK inhibitor protein called P21. So this will in turn activate P21. So this will now active. So this active P53 will bind to a regulatory region of the P21 now, because regulatory region of P21Y, oops, sorry, it also written like this, P21Y, because this is a regulatory protein. So for example, we will make a triangle of P21. So for example, this is P21, and this P21 is transcription gene. So it will have like this. This is, for example, the transcription. I will write it down here. Transcription P21. I think you can see that. Okay, maybe now. All right. So what it will do, it will activate this P21. Right, it active P53 binds to this P21, and it will become, now P21 will become active, like for this lines, maybe like a brown line. So this become active, and so this transcription translates to the, for example, this is transcription that translate to P21 RNA. So this is just in a bit detail, P21 RNA, for example, and that translates. So translation, then the translation will happen, and for example, this here we have our, how we made this thing, right? So it's like this and like this. For example, this is our active CDKs, right? active CDK is right here. I will write it down, active. Right now the CDK is active. So what will happen? This P21 will now inactivate this. So for example, this is our active one here, active CDK, and this will now attach or hold something like this. So this is our P21. Oops, again. P21, and this is our inactive CDKs. So this is how it inactivates our CDKs, right? Right. So once this is active, if it's active, so it will go from G1 to S phase. But once this CDK is active, like G1 CDK or S CDKs, they become inactive. The cell will be in the G1 phase, and it will not go to S phase until the DNA is repaired, right? And if the DNA damage is too severe to be repaired, what it will do, so instead of the P53 activating this P21, the P53 can induce the cell to kill itself by undergoing a form of programmed cell death that is called as apoptosis, as you know, right? And if the P53, if this protein P53 is missing or defective, what will happen? The unrestrained replication of damaged DNA leads to high rate of mutation and production of cells that tend to become cancerous, right? So in fact, the mutation in the P53 gene are found in about half of all human cancers. This gene, P53, this is, if it's mutated, and this is found in half of all the human cancer, the P53 is mutated. Alright, so as we discussed, like cell can delay progress through the cell cycle at specific transition point to wait for suitable conditions or to repair damaged DNA, right? So these are some of the factors. They can also withdraw from the cell cycle for prolonged period, either temporarily or permanently. So the most radical decision that the cell cycle control system can make is to withdraw the cell from the cell cycle permanently. Why? So this decision has a special importance in multicellular organism. So many cells, like in the human body, permanently stop dividing when they differentiate. So in such terminally differentiated cells, what are types such as nerve or muscle cells, the cell cycle control system is dismantled completely. So this cell cycle control system is dismantled completely, that we discussed here, cell cycle control system. And genes encoding the relevant cyclins and CDKs are irreversible shut down, and all the CDKs are permanently shut down. So in the absence of appropriate signals, other cell types can withdraw from the cell cycle only temporarily, and it will go on entering an arrested state called G0 state. All this G0 state, like it's not G1 but it goes in the G0 state. So they retain the ability to resemble, but when the cell cycle is in the G0 state, it can activate and again can complete the cell cycle. So most liver cell, for example, most liver cell are in the G0, cells are in G0 state. Most liver cells are in the G0 state, but they can be stimulated to proliferate if the liver is damaged, right? So most of the diversity in the cell division rates in the adult body lies in variation in the time that cell spends in G0 or in G1. So some cell types, including liver cells that we discussed, normally divide only once every year or two, whereas certain epithelial cells in the gut divides more than twice a day to renew the lining of the gut continually. So like in our gut system, so the epithelial cell divides like twice a day, but in the liver it divides like every 1 or 2 years. So many of our cells fall somewhere in between. They can divide if need arises, but normally do so infrequently, right? So this is maybe the half part of the topic. So we discussed from G1, right? So we'll next topic we'll discuss S phase, and then we'll discuss the M phase, that is our mitosis, right? So I think that is enough for right now, right? We'll see in the part 3 of this video. Right, guys, thank you guys for watching, and don't forget to subscribe.